Hi, my name is Kole DeGolier. I'm a Ph.D. candidate in the Immunology graduate program at the University of Colorado Anschutz Medical Campus. I'm in the laboratory of Dr. Terry Fry, and I'm going to talk to you today about how rational modifications to the CAR antigen binding domain can improve sensitivity to low-density target antigens. So chimeric antigen receptor, or CAR T cells, as most of you probably know, are a very effective therapy for leukemias and hematologic malignancies in general. On the left here, you can see that in this trial and others, that CARs have a 70 to 90% rate of remission induction when used to treat B-lineage leukemias. So in an anti-CD22 CAR trial led by Terry at the NCI, found that patients that relapsed often still had CD22 positive leukemia. And so this is in contrast to what had been seen in previous trials targeting, such as those targeting the CD19 antigen, where relapses were CD19 negative. So this trial, they saw that patients relapsed with CD22 positive leukemia. And on the right here, you can see that relapsed patients commonly had lower CD22 surface expression as compared to pretreatment levels. And so this really led to the hypothesis that these CARs need a high level of surface antigen expression in order to be fully effective, and this particularly against the CD22 target antigen. So our lab wanted to study this phenomenon, and so we've generated a number of leukemia cell line derivatives from the NALM6 human leukemia, and we've knocked out the CD22 target antigen and then reintroduced it at lower than wild type levels, surface density levels. And so on the left here, you can see that in the top of this figure, when CAR T cells are used to treat NALM6 leukemia on the right, they clear leukemia fairly well and prolong survival even at this lower dose. However, when they're used to treat 22 low leukemia, they're not as effective in clearing the leukemia. So in addition to this in vivo study, in vitro studies have been done that show that CARs have a number of functional deficits when stimulated by low antigen density, including lower IL-2 production. So our lab has studied this thoroughly with the CD22 target antigen found on hematologic malignancies. However, what's emerging in the field and what's been seen is that other hematologic antigens, such as CD20 and CD19, are also, CARs targeting these antigens are also affected by antigen density. In addition, antigens found on solid tumors, such as anaplastic lymphoma kinase, or ALK, on neuroblastoma, or EGFRV3, CARs targeting these antigens have also been found to be affected by antigen density. And so this lack of antigen sensitivity is a broad phenomenon of CAR T cells across target antigens and a major area for improvement in the CAR T cell field. So we wanted to go back and look at what parts of the CAR could be modified in order to enhance this response to low antigen density tumors. So in order to really analyze the structure of the CAR, we have to observe the receptor, which it is designed to somewhat emulate, which is the endogenous T cell antigen receptor, or TCR. So in contrast to the TCR, which recognizes peptide antigens presented on tumor cells in the context of MHC molecules, the CAR redirects T cell specificity using an antigen binding domain that's commonly derived from an antibody variable region, which is at the top of the CAR, and then this is connected to intracellular signaling moieties, which commonly include the co-stimulatory domain, such as CD28 or 41BB, as well as the CD3 zeta chain from the TCR complex. So some straightforward modifications have been made to the CAR that can have effects on antigen sensitivity or CAR performance. And These include the co-stimulatory domain, and so swapping this out has been shown to make a difference, as well as the number of immunoreceptor tyrosine-based activation motifs, or ITAMs, in the CD3 zeta chain, which are represented in red. However, recently our lab and others have made the observation that portions of the CAR construct previously thought to be almost inert can have large impacts on CAR biology. And one of these is the flexible linker, amino acid linker region between the heavy and light chain in the antigen binding domain of the CAR. And so a group at Penn made the observation and published recently that their CAR didn't perform as well in patients as the CD22 CAR from the trial at the NCI, and found that, astonishingly, one of the only differences was, in fact, the flexible linker domain that I just showed you. And so they wanted to study why this was the case. And so they found that they made a CAR with a short linker, and these linkers, flexible amino acid linkers, commonly have been constructed from motifs that consist of 4 glycine residues followed by a serine. So a short linker is just one of these motifs, while their long linker CAR had 4 of these motifs in between the heavy and light chain of the antibody. And they found that when they compared short and long linker CARs, one of the major differences was the long linker CARs are able to permit heavy and light chain pairing on an individual CAR receptor, whereas G4S1 short linker does not permit heavy and light chain pairing, and instead favors pairing of heavy and light chains from distinct CARs, which causes antigen-independent dimerization. And so in the figure on the left here, they show via fluorescent microscopy that the shortening of the linker length induces CAR clustering and antigen-independent signaling, as compared to the long linker CAR, which is more homogeneously distributed throughout the cell. However, they also found that when they compared these two CARs, that this antigen-independent clustering induced signaling as well, and tonic signaling of multiple TCR signaling pathways, including PI3 kinase, MAP kinase, and P53. So we hypothesized, based on the trial, on Terry's trial, that another important factor with this short linker CAR was not only the ability to do these things, but that this would enhance the ability to respond to lower antigen densities seen in patients treated with CD22 CAR T cells. And so we hypothesized that this would require, that this target antigen in particular requires enhanced antigen sensitivity. So we wanted to test these, our short and long linker CAR T cells, against our range of leukemias bearing different antigen densities. And we found that in contrast to the short linker CAR, which was in the clinical trial, when we made a long linker CAR, that shortening the short linker CAR improved sensitivity to low density target antigens. And so especially when you look at the CARs targeting CD22 low leukemia, you can see that the short linker CAR has significantly higher numbers of cells making IL-2 and interferon gamma in response to a low antigen density stimulation. However, as anti-CD22 CARs utilizing scFvs derived from the M971 antibody clone bearing these short and long linkers have already been evaluated in the clinic, we sought to assess how other modifications to the antigen binding domain could affect CAR T cell function, and whether combining these modifications could further enhance CAR T cell responses to low density antigens. Our hypothesis is that affinity maturation of the CAR antigen binding domain will enhance responses to 22 low leukemia, and this will increase overall therapeutic efficacy of CARs targeting the CD22 antigen. So in order to in order to develop a CAR with a high affinity CAR, our collaborators affinity matured the M971 binder, and this resulted in an scFv with approximately 1,000-fold higher affinity for CD22. And you can see this on the left, the standard affinity on the top, the high affinity on the bottom. The high affinity binder has approximately 1,000-fold difference in the dissociation constant Kd. We also wanted a simple cell-based readout to ensure that when we developed these binders into a CAR, into the receptor, and transduced T cells, that we would see a similar change in affinity. So we transduced CARs into T cells with either the high affinity or the standard affinity binder, and incubated these cells with a range of concentrations of a fluorophore conjugated CD22 protein Fc. And in this assay, we can see enhanced CAR binding of CD22 with the high affinity binder, as indicated by the red trace, and as compared to the blue trace, and this is indicated by increased MFI at lower concentrations of target antigen, indicating that the higher affinity binder shows enhanced CAR binding. So we went on to test this in vitro, and in contrast to our hypothesis, we found that affinity maturation does not enhance CAR cytokine production in response to low-density target antigens. And in fact, when you compare the top row here, the high-affinity CAR across antigen densities, to the standard affinity CAR, we found that in general, the high-affinity CAR had lower numbers of cells making either IL-2 or interferon gamma, in contrast to the standard affinity CAR. We wanted to go on and test this a little bit further, and so we looked at different signaling molecules that are known to be important in T cell function. So again, we found that affinity maturation does not increase the percentage of CARs that phosphorylate essential T cell signaling proteins. Phosphorylation of the extracellular signal-related kinases, ERK1 and 2 in particular, have been previously identified through SILAC analyses in our lab as highly correlative with CAR T cell in vivo activity. So when we looked at phospho-ERK on the top, we found that across antigen densities there were fewer high-affinity CAR T cells phosphorylating ERK as compared to the standard affinity CAR. We also looked at several other signaling moieties known to be important for T cell function, and these include NF-kappa B, PLC gamma, and P38 MAP kinase. And again, we saw that there really was no difference in these signaling moieties between the high affinity and standard affinity CAR. So then we took these CARs and wanted to do in vivo testing against our 22 low leukemia. And in contrast to the in vitro data, we found that the affinity matured CAR enhanced in vivo responses to CD22 low leukemia. So on the left is radiance data or bioluminescent imaging data comparing the standard affinity and high affinity CARs with luciferase labeled leukemia. And we found that the high affinity CAR significantly increased early leukemic clearance compared to the standard affinity CAR. And this also manifested in the increased survival of mice that were treated with high affinity CARs as compared to standard affinity CARs, as you can see on the right. So what I didn't mention was that this high affinity scFv was sent to us containing a G4S/3 linker sequence, which is, as I mentioned, a longer linker that does not permit CAR dimerization on the cell surface. So after understanding the biology of this response further, and seeing this promising in vivo data with the high affinity long linker CAR, we then hypothesized that combining modifications to the CAR antigen binding domain, in particular this affinity maturation and shortening of the linker length, would have synergistic effects on CAR sensitivity to low-density target antigens. However, what we found was that when we combine these modifications, these two discrete modifications of the antigen binding domain are independent in boosting in vitro CAR responses to CD22 low leukemia. So we tested the high affinity long linker on the top, the high affinity short linker in the middle, and standard affinity short linker on the bottom. And while we saw that in this donor, the high affinity long linker actually recruited more cells into the activated pool, producing IL-2 or interferon gamma compared to the standard affinity short linker, the high affinity short linker CAR performed worse than both other CARs in this assay. We also looked again at phospho-ERK and again found no significant difference between the three CARs in number of cells phosphorylating ERK against 22 low leukemia. However, we obviously wanted to test this CAR again in vivo and see if it improved leukemic clearance in xenograft models. And what we found was that although the high affinity short linker CAR perhaps performed slightly worse in vivo, that the regardless of linker length, the high affinity CARs performed very similarly to each other in vivo, and both high affinity CARs outperformed the standard affinity CAR in clearance of CD22 low leukemia. And so this can be seen again by bioluminescent imaging data on the left, in that these two CARs both have very similar early leukemic clearance relative to mice treated either with mock T cells or standard affinity CAR T cells. And we also looked at survival of these mice and found that both high affinity CARs significantly increased survival relative to the standard affinity CAR, and that both the high affinity CARs were very similar in their increase of survival. So in conclusion, affinity maturation of the antigen binding domain does not significantly enhance in vitro CAR responses to CD22 low leukemia. However, when we tested the affinity matured CAR in vivo, we found that the affinity matured high affinity CAR outperformed the standard affinity CAR in clearance of CD22 low leukemia. When we combined the modifications to the antigen binding domain, including shortening the linker and increasing affinity of the binding domain, we found that these discrete modifications that individually enhance CAR function are not always synergistic in their increase in CAR functions against low antigen density leukemia. And so further study of methods to enhance CAR efficacy in the context of low-density antigens is warranted. And we have manufactured and are into testing other CARs with short linkers, and this includes one targeting the AML-associated antigen CD33. However, in order to further test this phenomenon, I've generated using a syngeneic mouse model to study the in vivo responses of CAR T cells, and particularly the immune biology in response to low-density target antigens. So this system consists of three parts. We have a murine B lineage leukemia, B lineage acute lymphoblastic leukemia, or BALL, and this is on the same genetic background as our immunocompetent mice, and murine CAR T cells from genetically identical donor. And so these can be both syngeneic or adoptively transferred into a syngeneic host, and we can study the immune biology of CAR responses to leukemia in this host. Other advantages, though, include that we can control both the host environment and the presence of endogenous target antigen as well as different donor T cell intrinsic factors using the multitude of transgenic mice that are available to do so. Additionally, in the xenograft models, xenogenic GvHD can be observed and is commonly observed at late time points in these models. So as this model allows for the transfer of T cells in leukemia into a genetically identical host, this system extends our ability to study T cell persistence to much longer time points. And persistence has been shown to be a very important factor in CAR T cell responses in patients. So what we've found in preliminary studies in this model system is that as we see in the human system, that low target antigen density also impairs the function of murine CAR T cells. So as I've said, we've generated a number of leukemia cell lines of which express different antigen densities by knocking out the CD19 target antigen and reintroducing it at lower than wild type levels. And so we can study the responses of CARs targeting these different cell lines. And when we look at in vitro interferon gamma production, we see that the murine CAR T cells, very similarly to human CARs, are affected in the number of cells capable of producing interferon gamma in response to low antigen density stimulation. And we very much look forward to using this model system to further interrogate the in vivo biology of CAR T cells responding to low density target antigens. So I would like to thank the Lab Roots organizers for having me today. I would also like to thank the Fry Lab, particularly Terry, as well as Zach Walsh, a previous member of the lab who generated some of the data I showed you today. I'd like to thank the Kole Lab, particularly Eric, for his help and input on this project, as well as Terry's funding sources. Thank you very much.